All right, we are going to start the scary stuff. Don't worry, though, I'm going to break it down for you, but cellular respiration is definitely not one of the easiest topics that we will cover in this class. And this is difficult because it involves energy, it involves some chemical reactions, and also there's a lot of different levels at which we can study these processes. The thing I need you to keep in mind is that you are not biochemistry majors in college yet. Okay, so as we go through these slides, try to keep that in mind and try to relax and just kind of take in what it is that you have to know for this. And this is our first video on energetic reactions. And so I'm going to put some intro information here at the beginning. It's not too difficult. And then we'll go into glycolysis in this first video. So we'll go into the first step of cellular respiration. So let's get started. Energy, just on this slide, just a quick recap of what we should understand about energy. You should know at least kinetic, potential, chemical, and thermal energy, right? You probably had those in a physical science class of some sort, whether it was chemistry or physics, or you may be taking it now. But, kinetic energy is relative motion. Usually when we're talking about it in biology, we're talking about the motion of molecules. Thermal energy is basically heat, and usually heat is a measure of kinetic energy as well. Potential energy, usually, again, we think of this in physics as the energy of position, right? That things that are, for example, higher off the surface of the earth has greater potential energy. But when we're talking in biochemical terms, potential energy is really the energy that something possesses because of the position or structure. Usually we're talking about molecules that, and again, that's going to relate to the chemical energy, right? Which is the energy that is available in bonds for when we break or create bonds. And then there's this concept called free energy. And free energy is the energy that's available to perform work in a system when temperature and pressure are uniform. And free energy, for example, would be a lot of the energy that we use in these energetic reactions to get them started, like photosynthesis, for example. A lot of the free energy is coming from sunlight, right? So you're using that energy. All right, so just a quick review of that, right? So there's potential energy that's being converted to chemical or to kinetic energy, excuse me, as the diver dives down. So that's just a quick potential and kinetic energy. Now at the lowest point, right before the diver hits the water, he or she has the greatest kinetic energy, but in the water they have the lowest potential energy. But that energy gets converted, that kinetic energy gets converted to potential energy as a diver climbs up the platform. In other words, what this is leading us to is our laws of thermodynamics, right? Energy is constant, so it's neither created nor destroyed, but it can be converted as it moves through a system. Usually in biology, that's movement through being passed, like through a food chain or something like that. And that certainly relates here, right? Because we're talking about cellular respiration. So we're talking about breaking down molecules, usually from food, right? But understand that energy transfer is not efficient. That little can be converted from one form to the other is a bit of a caveat there. That's explaining that no energy transfer is completely efficient. Some of the energy is going to be converted to a form that we don't really want it to be. So we take in a bunch of chemical energy and we'd like to convert that chemical energy into other forms of chemical energy. But sometimes that energy gets converted into thermal energy, gets released as heat by our cells, which doesn't help us to do the jobs of the cells, but it does give us our constant body temperature. The second law of energy says that every energy transformation in the universe increases the entropy or randomness or disorder in the universe. But biological processes specifically attempt to counteract entropy and to create order where there is none. Okay, so these chemical processes that we're doing are meant to put back some of the order that we get when entropy increases. All right. So again, here's the first law of thermodynamics, right? Energy from the fish is being converted into chemical energy inside of this bear. But again, none of that heat is being efficiently transferred. Some of it is going to go as heat. Some of it is going to go in the form of these disorganized products like carbon dioxide and water. There's bond energy there, but it's not the same energy that the bond energy in the fish originally had. So there's a lot less bond energy. So you have less energy, more disorder, more entropy there. So first law, energy can be converted, but it's never lost. Second law says, as that energy is transferred, you're going to get more and more disorder created, unless, of course, our biological systems put back that order. All right. So in terms of capturing and storing free energy, autotrophs fall into several categories. The one you're most familiar with is the photosynthetic autotroph, right? That can capture energy from sunlight. But there are also, don't forget, the chemosynthetic organisms that are able to use hydrogen sulfide instead of light. The hydrogen sulfide that's in the hydrothermal vents at the ocean bottom, and can actually break the bonds in that hydrogen sulfide and use that energy to catalyze reactions very similar to photosynthesis. Heterotrophs generally metabolize carbs, lipids, and proteins for energy. And you remember from biochemistry that the primary source of energy that gets metabolized is carbohydrates. However, lipids have actually a lot more energy per gram than carbohydrates. They are harder to break down, so we don't use them as much. Plus, carbohydrates are immediately usable by all cells in our body. Glucose is able to be metabolized by any cell in the body quite easily. Lipids have to be transformed for us to be able to metabolize them. Proteins, same thing. Okay. And then you also have some heterotrophs that do cellular respiration, but they do a different form that ends in a process called fermentation. And those heterotrophs can metabolize. Their metabolism is not as efficient. The difference is that when they do their form of cellular respiration, their anaerobic form of cellular respiration, even though the products of fermentation aren't useful to them, they are still getting energy from the food that they're eating, while not as much as if they could do it aerobically, but anaerobes can actually still get energy in a low oxygen environment. So it's kind of a way of adapting to low oxygen environments, even though it's far less efficient than doing aerobic respiration. All right. So metabolism is meant to manage the energy and resources of the cell. So again, this is breaking down and building up molecules, and again, typically that rebuilding of molecules is what we think of as putting back the energy, right? That is as entropy builds up, it kind of reestablishes some of that order. So complex molecules can be broken down, converted into different products, and then simpler molecules can be built from those large, more complex molecules. Some molecules, though, have potential energy, again, because of the arrangement of their atoms. So some of that chemical energy is really because it's positional energy. It's position of the electrons that gives it its energy, which it gives us a form of potential energy. When we break these molecules down, that chemical energy or that potential energy gets released. Every metabolic process we're going to talk about involves electron transfer. And you'll recall from chemistry, when you have electrons moving from one molecule to the other, we call those redox or oxidation reduction reactions, right? So whatever accepts an electron is reduced. It's weird, I know, right? Because to accept makes us feel like we gained something, right? But you gained a negative charge, which means your charge is dropped. Okay, your charge is -1 when you accept an electron. The electron donor, however, has lost a negative charge and therefore it's oxidized. in the chemical processes we're going to see, sometimes you don't actually completely have to remove the electron for this to still be a redox process. Sometimes what happens is electrons in polar molecules might move closer to one atom in the molecule or to the other atom in the molecule. So let me show you an example of that here. Okay, so you've got carbon dioxide and water. Water here is a polar molecule, right? And actually, this is carbon dioxide is considered to be nonpolar because of its geometry. But notice the electrons here that are shown are closer to the oxygen, right? Here, same thing. But now look, this carbon dioxide gets converted through this reaction into glucose. But notice that now, as a result, when this oxygen gets pulled away and a hydrogen replaces it, the electrons move closer to the carbon. So the carbon actually draws that negative electron, those negative electrons toward it more often, which means that the carbon is actually reduced. And in this case, the oxygen that loses these hydrogens is bonded to other oxygens to create oxygen gas. And this is the basic rules of chemistry, right? When you bond an atom to itself, it's a nonpolar covalent bond. Both atoms in this particular molecule have the same pull on the electrons because they're both the same atom. They have the same electronegativity. And so now the electron is moving away from each oxygen individually and is being shared more equally between the two, which means the oxygen is oxidized. All right. So it's time to meet the players in these reactions that we're going to talk about. And this guy that you're seeing on your screen is pretty standard, right? We talked about it in AP Bio. What you see here is ATP, right? ATP consists of an adenosine unit, which is ribose sugar and adenine. And then attached to that are three phosphate groups. And remember, phosphate is one of those functional groups we learned about back in the biochem unit. So these are inorganic, but the adenosine unit is organic, okay? And when we take an ATP molecule and combine it with water or hydrolyze it, remember that from the biochem unit, right? We pop the phosphate off. Now this is an inorganic phosphate. We have what we call a DP. Remember, this is adenosine. The T stands for tri in adenosine triphosphate. Now it's adenosine diphosphate because now this ADP only has two phosphates on it. You have this phosphate group that is floating around. And depending on the textbook, my slide right behind here says you have 7.3 kcals. It got cut off, but kcals of energy or kilocalories of energy. Some textbooks will say 7.6, some will say closer to 8, but somewhere between 7.3 and 8 kilocalories of energy is released. So in other words, the energy that's in the bond that holds this last phosphate on right here, and it's really this bond right here, the energy in that bond is worth 7.3 to 8 kilocalories of energy. All right. We also have electron carriers that are going to be involved in cellular respiration. And there are two. There's one mentioned here, and then if you understand this one, you'll understand the other. The first one, and the one that becomes immediately important in glycolysis is NAD+, and it stands for nicotinamide blah, blah, blah. I don't remember, okay? Just it's always referred to as NAD+. And so the point of these, whenever you see the ADs, right? NAD plus FAD is going to be another one that you're going to see in the Krebs cycle. These are electron carriers, okay? What you have to understand about cellular respiration is it's a big process that has a lot of setup for a grand finale, but that grand finale is going to involve a lot of electrons. Okay, so these are going to be important for carrying electrons, and they do so by binding to hydrogen ions that can then pick up and attract electrons, okay? And why hydrogen? Because hydrogen's tiny. We can bully it. We can beat it up and take its electrons away, and then we can give them back when we feel like it, right? It's real easy to take an electron from a hydrogen and to put it right back on when we want to. So we can kind of use that to move energy around for us in these processes, okay? So we like to use we use hydrogens in these processes, and they're usually attached to one of these electron carriers. If you want to know what it is, it's actually a coenzyme, okay? And it actually can temporarily accept hydrogens. It can temporarily accept oxygen. It can temporarily accept electrons. But as the hydrogen gets stripped from glucose in glycolysis, we're going to be able to take these electrons, transfer them to NADH or NAD plus and create NADH. And again, the whole point in cellular respiration is to do a whole bunch of work at the beginning to build up molecules that we can set into motion in the last step, which will generate mass quantities of ATP for us. All right, so here it is, right? Here's the NAD plus, okay, and it's got two of these. Here's basically a nucleotide here, right? Here's a phosphate, a sugar, and a base, and that's ribose sugar attached to another phosphate sugar unit, and then the nicotinamide portion here. And again, it's taking hold of the hydrogen, and it forms NADH, okay? So NADH is the form that it's in when it's accepted electrons. And notice that's because it's accepted hydrogen ion and 2 electrons. All right, also we have to talk about the fact that in cellular respiration, you're going to see 2 kinds of ATP synthesis. You're going to see substrate level, sorry, I started to say phosphate level, substrate level phosphorylation, which is what you're seeing on this slide here. Essentially, you have one enzyme and it's working like mad to take an ATP and some phosphorylated substrate and stick that inorganic phosphate onto it to create ATP. This is a really inefficient form of creating ATP, and it exists in some parts of cellular respiration. The parts of cellular respiration that lead up to the grand finale are all going to be this substrate level phosphorylation that you're seeing here, okay? When we get to the grand finale, we're going to see a different kind of phosphorylation. We're going to see something called oxidative phosphorylation. And in that, it's a process that involves something called chemiosmosis, but I don't want to give the end away too much right now. But essentially, it's a really, really efficient form of phosphorylation that's going to turn out a bunch of ATPs for us in a very short period of time. Okay, so this is the overview, okay? There are 3 steps, but I always say there's 3.5 steps to cellular respiration, okay? The first step we call glycolysis, okay? And glycolysis takes place in the cytoplasm. And essentially, glyco refers to the sugar, like glucose. Lysis means to split, right? So literally, this is the sugar splitting step. We're going to split the sugar in 2 to create what we call pyruvate. Okay, so the half sugar molecules that we create through this process, we're going to call pyruvate. Then the half step occurs right in here, starting at this word which. This is often referred to as a link reaction. It's a step that doesn't actually generate any energy for us, but we need to do this. This is a link reaction that takes the pyruvates from glycolysis into the Krebs cycle, and that's the little half step that I like to talk about when I say it's a half step, to form something called acetyl-CoA. So we're actually going to attach the pyruvate to a coenzyme called coenzyme A to create acetyl-CoA. So acetyl-CoA is really pyruvate plus this coenzyme. That's going to lead us into the Krebs cycle, also known as the citric acid cycle, because one of the first intermediates that you create is citric acid, okay? And each acetyl-CoA will go through the Krebs cycle one time, but remember, we start with 1 glucose, we've now split it in half. So we actually do the Krebs cycle 2 times for every glucose molecule, okay? And this is going to take place in the mitochondrial matrix. In fact, what happens is during the link reaction, the coenzyme A, the pyruvates, get pulled into the mitochondria and they get converted in the mitochondria and shoved into the mitochondrial matrix. Now, like I said, these first 2 steps, you think, okay, cellular respiration, yeah, energy, that's what we're going to create from this, right? That's what we want. We want energy. They actually don't make that much energy. Remember, they're doing substrate level phosphorylation, okay? The big dance, the big energy comes from the last step. In fact, in the Krebs cycle, the sugar that we've been using to generate our energy and our electron carriers, that's gone. The sugar is completely metabolized away by the end of the Krebs cycle. But what we have left that we've created through these first 2 processes are those electron/hydrogen carriers, the NADHs, the FADH2 that you're going to see coming up. And those are going to be assembled into a magical process called the electron transport chain. And this is where we're going to get that oxidative phosphorylation, where you're going to generate a big bunch of ATP at one time. All right. So this is just kind of showing you an overview. Now, what's really nice and what I want you to think about as we do this is what's going in and what's coming out. Where are we and what's the point of these processes? Okay. And what you're going to see when we do these is a lot of sketchy diagrams. We're not going to be doing a lot of detail here because, again, you're not majoring in biochemistry, right? You just need to get the gist of each of these processes and need to understand what did we put into it, what are we getting out of it, and why did we bother with these steps, okay? So glycolysis is the first. Pyruvate processing that you're seeing here is that link reaction I was talking about. That's the thing I call the half step. The Krebs cycle, and then oxidative phosphorylation at the end. So that's cellular respiration as a whole.